So as an intro to genetics, some important words that we should be aware of are words like phenotype and genotype. So our genotype is basically the combination of alleles that we inherit, and they code for our phenotype. Now our phenotype is like our physical expression of those genes. So it can be like in our like our actual physical appearance, like brown eyes or freckles, but it can also be like on the inside that you can't actually see. So for example, the presence, like you having cystic fibrosis or not, could also be considered phenotype. So it doesn't have to be something you can actually see, but that's an easy way to remember it. Now for our genotype, we have chromosomes, and from our chromosomes we got one set from our mom, haploid, and then we have one set from our dad that makes us diploid. So we actually have two combinations, or two copies of each trait, one from one parent and one from the other parent. We call these homologous chromosomes. Okay, so on our 46 chromosomes, we have about 25 to 30,000 genes, and for each gene, we have one from our mom, one from our dad, and we call those things alleles. Now, when we actually break down our chromosomes and classify them, you can see here chromosomes 1 through 22 are considered to be autosomes. So when we talk about modes of inheritance, we can say things like brown eyes is inherited autosomal dominant, or we could say like, I don't know, blue eyes is inherited autosomal recessive. Or when we look at our sex chromosomes, for example, colorblindness is inherited on the X chromosome. So you would say colorblindness is sex-linked recessive. So when we talk about how traits are inherited, they're either inherited on autosomal chromosomes or sex chromosomes. And that's significant because for male and female, we have different sex chromosomes. Females are XX and males are XY. So in this karyotype here, you can see how it is a karyotype of a male because it has an X and a Y. So when you read problems for genetics, pay attention if it's talking about autosomal inheritance. That means that everybody has 1 and 2, like 2 copies of that trait. Versus if it's sex-linked, if you're a guy, you only have 1 X. So if on the X chromosome, you only get 1 copy of it. And that's why sex-linked versus autosomal is a little different. Okay, so you have 1 chromosome from your mom, and 1 from your dad, and you have genes on these chromosomes. So here may be the gene for eye color, and you can see how this is, you have like your dominant allele, and you have your recessive allele. Like we call these different versions of the same gene alleles. So the gene we're dealing with here is eye color. That's the trait or the characteristic we're talking about. Now you have 2 alleles for each trait. You inherit 1 from your mother and 1 from your father. Now you can have different versions of that. So this question here, how many copies of each gene do we have? We have 2 copies, 1 from each parent. So when we look at, though, how we inherit this, if 2 are the same alleles, you can have 2 different alleles. And so the words for this are homozygous dominant, homozygous recessive, and heterozygous. Okay, so when we look at this, though, remember in meiosis how in S phase, our chromosomes duplicate, and then during meiosis, the homologous pairs separate. So right here, this is key. You can see how the homologous pairs have separated. So now half of the gametes are going to end up with the brown eye allele, because here in the second meiosis number part 2, the second round of division, these sister chromatids separate. And so now you have 4 gametes, and you can see how half the gametes have the dominant allele and half the gametes have the recessive allele. So again, this is really important. So here you are, if you're this middle one that we're talking about, you've inherited a dominant allele and a recessive allele. You're heterozygous. So now in meiosis, these are homologous pairs pair up, and then they separate. And so in the first round, here you have haploid cells, but there's too much DNA, so they're going to separate again. Now this is Mendel's law of segregation. Alleles separate during meiosis. now you can see the gametes either have the dominant allele or the recessive allele. They don't have both. And so they're going to inherit only one copy, either dominant or recessive. And so when we do genetics and we do Punnett squares, what we're talking about is how these alleles have separated. We're looking at the possibilities of gametes. So in the Punnett squares, we put all the possible gametes on the outside of the box. Now this Punnett square, this is a tool for predicting probability. So when we fill in these boxes, what we're really saying is if this sperm fertilizes this egg, here's what the offspring will be. These are the possibilities of the offspring between these two particular parents. So the Punnett square is just a probability thing. And then we now can take that and apply it to Punnett squares. So when you're reading genetics problems, you're going to always want to write down what is dominant and what is recessive as you read. Right here is a very simple problem, but over time this is going to get more and more and more difficult. And so right away, having thorns is dominant to no thorns. I use the capital letter for thorns and I use the lowercase letter for no thorns. And so when we talk about genetics, we talk about like crosses, and crosses are talking about two parents having offspring. And so we can say here, we would call this, because this is a capital and a lowercase, we call this genotype heterozygous, because it has two different alleles. On the chromosomes, if I had a chromosome from the mom and a chromosome from the dad, this mom would have a capital A here and a lowercase here. And then the dad, homozygous recessive, so on these two chromosomes, it would be a lowercase a and a lowercase a. So the genotype for the mom is heterozygous. Those are the alleles that she has. And the genotype for the father is homozygous recessive, because those are the alleles that he's inherited from his own parents. So now when we talk about a cross is mating these two individuals. Now, this mom's phenotype is the actual physical expression of the traits. So that would be thorns, because thorns is dominant. And you actually only need to have one copy of a dominant allele in order to express that trait. So when we go to use a Punnett square, we're going to take these mother's possible alleles. So half her gametes will have a dominant allele, and half her gametes will have a recessive allele because of Mendel's law of segregation. Now, in the dad, all of his gametes have the recessive allele. He does not have any dominant alleles, so he will only contribute recessive alleles to his offspring. So when we fill in these boxes, like in this box number 1 right here, what we're going to do is we're going to put this A. It's like kind of like what happens if these are gametes. What happens if this particular sperm fertilizes this egg? Well, that's what we write in here. This offspring, we can see here, will be different. So when we look at these offspring, these two parents will have about a 50% chance of having offspring with thorns and a 50% chance of having offspring without thorns. So this doesn't mean that if they have 4 kids, 2 of them absolutely will have thorns and 2 of them absolutely will have no thorns. What this means, it's a probability. Some will and some won't, and it's about a 50-50 shot. And so when we look at, we can like further this along, and we can see here that when we talk about like a phenotypic ratio, well, 2 have thorns and 2 don't. So it's a 2-to-2 ratio, or we can do like a 1-to-1. And that's the same for genotype. You have heterozygous and homozygous recessive. Okay, so if we always write down what is dominant and what is recessive. So here you have the mom her phenotype is straight thumbs because she has dominant alleles. So a straight thumb, like if you do your thumbs up, go like this, thumbs up is straight thumbs, and then this is curved thumbs. So this one's better. So here's a straight thumb, and there's a curved thumb. So here, this dad's phenotype is straight thumbs. The mom's genotype is homozygous dominant because she has 2 dominant alleles. And the dad's genotype is homozygous. Is heterozygous. It has 2 different alleles. So when you do this Punnett Square, you put the possible gametes on the outside. And then as you fill in the Punnett Square, this is showing the possible offspring that can exist. And when we see this, we see for the phenotypes, they have 100% likely of having a kid with straight thumbs. That's the only possibility, and that's simply because this mom is homozygous dominant. She will give a dominant allele to all of her offspring, and therefore, to the end of time, all of her babies will have straight thumbs. However, the genotypes are 50-50. Half the kids will have homozygous dominant, and half the kids will inherit heterozygous. So when we do the ratios, you can see here for genotypic ratio, it's a 1-to-1. And for phenotype, it's not really a ratio because it's 100% will have that phenotype. Now in grasshoppers, you have short antennae are dominant. In grasshoppers, short antennae are recessive. So then the opposite of short would be long. And so here you can see how I've written down capital A is long, lowercase a is short. And here I have a mom whose phenotype is long antenna and whose dad's genotype is heterozygous, phenotype is long antenna. So here I have 2 long antenna parents who are both heterozygous. When we do their Punnett Square, we can see that they have a 75% chance of having a kid inheriting long antenna as its phenotype. So here the phenotypic ratio is for every 3 long antenna, there's like a 1 short antenna. So 75% probability versus 25% probability of short antenna. So we write this phenotypic ratio as a 3-to-1. And then in the genotypic ratio, you have here 3 different genotypes. So the way we write this is a 1, which is here, 1 to 2 to 1. And so the genotypic ratio would be that. Okay. And just some other vocabulary when we talk about genetics. You have true breeding. True breeding is purebred, or homozygous dominant, or homozygous recessive. And then when you cross these two, though, all of the offspring are heterozygous. We call that hybrids. So we also call the first generation the F1. So this would be the parental generation. This would be the F1 generation. And then if we let those cross, you have the F2 generation, which is kind of like grandkids. And you can see here we have that recessive phenotype. We also see that 3-to-1 ratio of 2 heterozygotes crossing. Now these are not true breeding, so you can see here it asks, are these true breeding? No, these are considered heterozygotes or hybrids. Cool, good job.